In this video, we'll discuss ADH, antidiuretic hormone, or arginine vasopressin. Vasopressin is a nonapeptide like a oxytocin and has a disulfide link between the position 1 and position 6. What's the difference in the structures of oxytocin and vasopressin? The leucine in oxytocin on position 8 is replaced by arginine in vasopressin, and also isoleucine on position 3 is replaced by phenylalanine in vasopressin. So what's the difference between the synthetic analog of vasopressin with the ADH? The synthetic analog has the structure similar to the vasopressin with a disulfide linkage. The desmopressin, the synthetic analog, has high antidiuretic activity with little pressor activity. Desmopressin differentiates between central and nephrogenic diabetes insipidus. Desmopressin increases urinary osmolality by at least 50% in central diabetes insipidus but not in nephrogenic diabetes insipidus. Where ADH is produced. ADH is produced in the ribosomes of the magnocellular cells and packaged in the Golgi apparatus or herring bodies of the supraoptic and paraventricular nuclei of the hypothalamus. It is also produced in brain and spinal cord. So how does it go to the posterior pituitary? It travels through the hypothalamo-hypophyseal tract to reach the posterior pituitary gland and is stored there until released. How ADH is released? Angiotensin II act on the subphonical organ and organum vasculosum of the lamina terminalis to increase ADH secretion. Hypothalamic osmoreceptors are stimulated by increase in plasma osmotic pressure of more than 290 milliosmoles per kg or a 1 degree change in osmolality. The action potential is generated that reaches the axon terminals in the posterior pituitary and causes the release of hormone by calcium-dependent exocytosis. What's the average set point? The average set point for ADH release is 280 milliosmoles per liter of plasma osmolality and 135 milliequivalent per liter of sodium concentration. The set point is lowered in pregnancy, menstrual cycle, and estrogen. What's the effect of chronic stimulation on set point of ADH secretion? Chronic stimulation of vasopressin secretion, as in hypovolemia, lowers the threshold for vasopressin secretion or the set point, leading to water retention and decreased plasma osmolality and water intoxication. So what are the precursors of ADH? The precursor is presophysin and is associated with neurophysin inside the granules. The half-life is about 20 minutes and the action is quick and short-lived. Where ADH acts? Mainly ADH acts on the collecting ducts of the renal tubule and causes retention of water in excess of solutes, reduces the effective osmotic pressure of the body fluids, concentrates urine and increases the urinary specific gravity. The cortical and inner medullary collecting ducts absorb 5% of sodium. Water and sodium balance are regulated independently. Water by ADH and sodium by the aldosterone. So what are the types of cells in the collecting ducts? There are two types of cells in the collecting ducts: the principal cells and the intercalated cells. Let's discuss the principal cells first. ADH acts to cause water reabsorption in the principal cell. The cell membrane have water channels called aquaporin, which are protein in nature. So what are the types and locations of the aquaporin channels? Aquaporin 2, 3 and 4 are on the collecting duct. The aquaporin 2 is on the apical surface, and aquaporin 3 and 4 are on the basolateral surface, whereas aquaporin 1 are on the proximal and distal tubules. And where are the ADH receptors? V2 receptor, vasopressin 2 receptors are present on the basolateral surface of the collecting ducts, whereas V1A or V1 is present in the smooth muscles, platelets and liver cells, whereas V1B or vasopressin type 3 receptors are present in the anterior pituitary. So the AVP acts on the vasopressin 2 receptor on the blood side or the serosal or basolateral surface of the collecting duct, and it activate adenylate cyclase through a G protein mediated cascade to produce cyclic AMP, and send signals to insert water channels aquaporin 2 on the apical surface to increase the permeability of water to enter into the cell. So the water is absorbed passively from the lumen of the collecting duct into the cells, and then absorbed into the hypertonic medullary interstitium through the aquaporin 3 and aquaporin 4 channels. Cyclic AMP also increases the permeability to urea and some solute. What are the parts of the nephron which are impermeable to water? The thick ascending limb of the loop of Henle. Number 2 also it's the distal tubule. And number 3, collecting duct in the absence of ADH. Only active reabsorption of sodium and chloride occurs in thick ascending limb and distal tubule. Water is not absorbed there. The other functions of the ADH. ADH acts on the V1 receptors in the smooth muscle blood vessel, causing arterial vasoconstriction, and in large doses increases the blood pressure, and it also decreases blood flow in the renal medulla due to vasoconstriction. ADH also increases the permeability of the collecting duct to urea and other solutes. Acting on the V1 receptor present on the platelet and hepatocytes, it causes platelet aggregation and glycogenolysis in the liver. Acting on the V1B receptors or V3 receptor on the anterior pituitary, it causes ACTH release. What are the effects of ADH absence? Urine becomes hypotonic to plasma. There is polyuria and polydipsia. So what are the stimuli that increase the ADH secretion, and which of them is the most potent stimulus for ADH secretion? The stimuli that increase the ADH secretion are increased plasma osmotic pressure, decreased ACF volume, hemorrhage, surgery. And nausea is the most potent stimulus for the ADH secretion. Nausea increases the ADH secretion by 50 to 100-fold, and this effect is blocked by antiemetic. Other factors that increase the ADH productions are acute hypoglycemia, glucocorticoid deficiency, and hyperangiotensinemia. The physiologic factors that increase the ADH secretion are exercise and smoking. What are the drugs that increase the ADH secretion? Morphine, nicotine and barbiturates, chloropropamide and carbamazepine. They stimulate the ADH secretion. How hemorrhage stimulates the ADH secretion? Hemorrhage is a potent stimulant for ADH secretion. The hemodynamic response is mediated by acute reduction up to 10 to 20% in the blood pressure or volume. It stimulates the ADH secretion by stimulation of the baroreceptors in the low and high pressure system via the glossopharyngeal and vagus nerve. Stimuli that decrease the vasopressin secretion are decreased plasma osmotic pressure, increased extracellular fluid volume, alcohol and butorphanol, which is an opiate antagonist. What are the non-osmotic factors that regulate the ABP secretion? They are nausea, stress, hypoglycemia, and pregnancy. When there is decrease in ADH in pregnancy, there is 3 to 4 increased destruction of ADH in the second and third trimester of pregnancy. And why? It is due to the increased production of vasopressinase from the placenta.